Hello, my name is Antoniya Aleksandrova, and I am a postdoc at Lucy's Forest Lab at NIH in the US. Today, I'd like to tell you about my work on membrane protein symmetry. And before I begin, I'd like to thank all the people I've listed here who have contributed greatly to this project throughout the years. And also thanks to the organizers for letting me share my work with you. But without further ado, why do we care about symmetry? There are many examples of how symmetry in membrane proteins is related to function and evolution. For example, channels transport substrates down their concentration gradient. Therefore, a common solution to this task is a structure that consists of units replicated around a central axis that is orthogonal to the membrane plane, as can be seen in the quaternary structure of the potassium channel shown here. In this way, you can have a pore through which the substrates can diffuse. On the other hand, secondary active transporters, which move a specific substrate against its concentration gradient, tend to have structures with symmetry axis that is parallel to the membrane plane. These types of repeated elements are called inverted or antiparallel repeats because of their opposite orientation with respect to each other in the membrane. The reason why these types of repeats are favored in transporters is probably connected to the fact that they can create a mechanism that is consistent with the requirement of an alternating axis transport cycle. So consider two inverted repeats. By creating an asymmetry in one of them, you can have an opening to just one side of the membrane. And by allowing the repeats to swap conformations, you can have an opening to the opposite side of the membrane. Alternating between these two states allows a transporter to carry a substrate across without dissipating the vital electrochemical potential that exists across the membrane. Given how interesting and functionally relevant these symmetries are, it seems worthwhile to quantify and systematically study them, but to do so, we need to be able to readily detect them. So how easy is it? Well, it might seem like a trivial task for structures that are perfectly symmetric, like the potassium channel, which has a perfect quaternary symmetry between its subunits or chains. It looks like a manageable challenge for the pseudosymmetric cases in which the repeats might have vastly different sequences, but very similar structures, as in the example of the ADP-ADP carrier shown here. But as the symmetries start to break down, or multiple different symmetries appear within the same protein chain, as in the glutamate transporter homolog shown here, GOTPH, this exercise becomes quite challenging. So can existing methods detect membrane protein symmetries? To answer this question, we built a benchmark dataset called MEMSTATS, which consists of 80 complexes and 140 internal symmetries with different folds. And we proceeded to test the most popular symmetry detection algorithms on it. When considering quaternary symmetries, that is, the symmetries between complex chains, we see that many quaternary symmetries are readily detected, especially by tools that are designed specifically for quaternary symmetries, such as ANANAS or QuadSym, which is the one used in the PDB. On the other hand, if we consider the symmetries within individual chains, or the internal symmetries, the best available tools, CSIM and SIMD, still failed in quite a few cases. For example, the internal symmetry of the glutamate transporter homolog GOTPH is not detectable by these tools. Furthermore, none of the symmetry detection algorithms tells us about the orientation of the symmetric repeats relative to the membrane, which, as you saw earlier, might be relevant. Therefore, to address questions related to the role of symmetry in membrane proteins, we needed a method that can better address some of these challenges. So instead of using the developed algorithms as were, we devised a multiple-step symmetry detection method around them. We take the PDB of a membrane protein and we insert it in the membrane using the tools behind the OPM database. We then use in tandem a couple of previously developed symmetry algorithms such as CSIM and SIMD, but with multiple custom parameters. And we also use information from one algorithm to improve the performance of the other. In this manner, we increase the possibility of detecting any symmetry at this step. To prevent us from producing many false positives, we then implement a filtering procedure, which discards results that do not seem to have functional relevance, such as symmetries that are external to the membrane, or a single helix matched to another single helix. Finally, we select the results that will provide us with the most symmetry-relevant information, such as the option with the maximum coverage shown here. So how does MSSD measure up to the other algorithms we tested? For both quaternary and internal symmetry detection, MSSD performs exceedingly well, as shown by the blue bars. We're now able to detect cases as difficult as the internal symmetry of the glutamate homolog, transporter homolog. And by design, MSSD provides us with the orientation of the symmetry within the membrane. Therefore, we proceeded to analyze all available membrane protein structures, and we built an automated online database to share our results. The first version of this database, called Encompass, was released in 2018. But recently, we underwent a series of major updates to allow us for a greater coverage of the available membrane protein structures. We removed previously imposed restrictions on the resolution and the experimental method used to solve the structure. And in addition, the growth of available membrane protein structures, which since 2018, there, according to Stephen Wise database, there are about 700 new unique membrane protein structures. All of this has led to a much larger Encompass dataset with about 3 times the size of the 2018 version. So before I go back to our discussion on symmetry, let me briefly show you what kind of structures can now be found in Encompass. To give you a more representative overview of the type of structures available, I will show you the data for a non-redundant data set that contains structures with less than 40% sequence identity. One measure of the size of the proteins is the number of transmembrane regions that a complex has. So plotting this, we see that the vast majority have less than 25 transmembrane segments, with peaks at 7, 12, and 24. 7 for GPCRs, 12 and 24 for transporters and channels with fourfold symmetry. Notably, though, the database now includes enormous complexes with over 200 transmembrane segments, such as these giants here, a type 7 secretion system from tuberculosis and a phospho-to-system 1 complex. Proteins like this can present challenges not only in terms of scaling, but sometimes in the types of global and local subsymmetries that can be detected. And are these complexes that we encounter mostly made up of big chains or many small chains? To see that, we have plotted the count of helices per chain, or membrane spanning regions, on the y-axis here, and the number of membrane spanning chains per complex on the x-axis here. So monomeric proteins and their transmembrane counts are along the first column, while every instance of a single transmembrane chain will be along the bottom here. And assemblies of large subunits would be up in the corner. So plotting it this way shows that individual chains rarely contain more than 12 transmembrane segments, but that there can be almost double this amount of subunits in a complex. In particular, we see that a lot of the really large complexes contain many individual subunits which span the membrane just once. We don't see many large complexes made up of large individual chains. From a structural symmetry detection point of view, this can be a challenge since it requires a reliable way of determining the exact structural order in which the chains are arranged. But it is also fascinating to consider the implications of this observation for assembly prediction efforts. As we saw, the updated Encompass dataset is more complex, but while this presents a challenge for the analysis we want to run, it also provides better statistical power since we have substantially more data. I started by telling you how important symmetries are, so what can we say about the symmetry patterns in the context of this new dataset? In our 40% non-redundant dataset, we have about 160 monomers and 500 multi-chain complexes. So does the membrane alter their predisposition to form symmetric structures compared to water-soluble proteins? We find that about 88% of the multi-chain complexes have quaternary symmetry, which is very close to what has been estimated for water-soluble proteins, as indicated by the white dotted line here. On the other hand, membrane proteins are about twice as enriched in internal symmetry compared to water-soluble proteins. And what about the orientation of the symmetry with respect to the membrane? Internal symmetries are about equally likely to have parallel or inverted repeats. Strikingly, this is not the case for quaternary symmetries. It is in fact exceedingly rare to find inverted or antiparallel repeats forming a complex. When you consider these findings in the context of what I told you earlier about the mechanisms of secondary transporters, this data makes it apparent that the functional unit of most transporters is the chain and not the complex. But can we quantify these putative symmetry function relationships further? Are there indeed specific symmetries that are associated with specific functions? To systematically address this question, we turn to the gene ontology database. While we explored other ways to link ENCOMPASS structures to functional orientations, the gene ontology database provides the largest coverage, so we'll focus on it here. As many of you know, the Gene Ontology Project, or GO for short, curates experiment-based gene annotations that describe the gene's molecular function, the biological process they contribute to, or the cellular compartment where the product is active. To connect ENCOMPASS structures to specific GO terms, we relied almost entirely on the SIFS project. And in very limited cases, when SIFS wasn't able to assign GO terms to structures, we also looked them up in the UniProt database. Here we present the results for a dataset that has once again been filtered to contain structures with no more than 40% sequence identity, and we're limiting it to alpha helical transmembrane proteins for simplicity. And the result is 746 complexes that are connected to 4,628 GO terms, and each protein is associated with at least one GO term. So finally, we analyze these results by calculating the enrichment of the GO terms within each type of symmetry and the significance of the enrichments using a corrected Fisher's exact test. We also make sure to remove redundant GO terms, which we define as ones that are shared between over 50% of the same structures. So do specific quaternary symmetry orders relate to specific functions? The largest group in our population is of proteins that have no quaternary symmetry, either because they're monomeric or because they're part of a non-symmetric complex. Here, I'm listing some of the top most significantly positively enriched non-redundant GO terms within the symmetry group, so within the no symmetry group, and in order to give you a flavor of the patterns that we see. So in particular, here we see an enrichment of macromolecule transmembrane transport activity and amide binding. The size of the enrichment is indicated by the circle size, which has been scaled to the maximum enrichment among all groups, which in this case is 30. So for the first term shown here, we are reporting that macromolecule transmembrane transport activity is 2 times more likely to be associated with no symmetry versus any other quaternary symmetry group. And the p-value tells us that the likelihood of seeing this enrichment by chance is of the order of 10 to the minus 4. So the proteins described by this GO term enable the transfer of a macromolecule from one side of a membrane to the other. These proteins tend to accommodate large substrates and might recruit and interact with other proteins in a dynamic fashion. The example shown here is of TAT-C, which is a part of a main pathway in thylakoid membranes for transporting folded proteins. The C2 symmetry the complexes in this group make up about a third of our data set and show modest enrichment of ion binding, adenyl-ribonucleotide binding, and active transmembrane transporter activity. Most proteins in this group have a binding site or pathway at the repeat interfaces. And this is the case for the ABC transporter example shown here, which uses the energy of ATP binding and hydrolysis to translocate substrates across the membrane. Next, we examine the less populated higher order cyclic groups. In the C3 symmetry group, we note a robust but mildly significant enrichment of enzymes that catalyze redox reactions that cleave peroxides, while C4 and C5 symmetries are noticeably enriched in ion channels. C4 and C5 symmetries allow for the creation of a pore through the membrane that can have a selectivity filter capable of coordinating an ion. And what about internal symmetries? Do they follow a similar pattern to quaternary ones? First, we know that once again, the most populated groups are the no symmetry and the C2 symmetry groups. Within the C2 symmetry group, we report a very significant threefold enrichment of secondary active transmembrane transporter activity, which, as per our discussion in the beginning, is a great example of the structure-function relationship. In fact, if we separate this whole data set into parallel versus antiparallel or inverted repeats, we see that the enrichment in the C2 group is due primarily to the antiparallel symmetry or the inverted repeats, because they are the ones carrying the secondary active transmembrane transporter activity functional relationship. Whereas in the parallel ones, we don't see them enriched. So, as I said, secondary active transporters are enriched in the C2 symmetry, but they also show up in the dihedral G2 symmetry. And however, here, the secondary active transporters are transporting larger peptides, and that probably is enabled by the flexibility that this dihedral fold allows. Finally, the internal C3 symmetry groups contains only 22 proteins, which show high enrichment in oxidoreductase activity. The proteins often form complexes with non-internally symmetric proteins, which is why the oxidoreduction-driven active transmembrane transport activity also shows up in the no internal symmetry category. There we also find mildly enriched transferase activity and cation binding. To summarize, today I highlighted three main observations from a study of the ENCOMPASS dataset, namely, the propensity for large complexes to be formed by many single transmembrane segments rather than big chains coming together. Also, the striking difference in the topology orientation when considering internal versus quaternary symmetries, and the clear relationship between certain protein symmetry groups and the protein function. We have done a fair number of other analyses with ENCOMPASS, and much more can be done. So I invite you to check out the database and reach out with any questions or comments. Thank you.